Hello, everyone. Thank you so much for coming. Welcome to my presentation on engineering novel systems to expand neural cryopreservation in supercooled conditions. Essentially, what we have developed is a new method of inducing a stable supercooled state in the commercially available cell culture vessels, allowing for the safe cryopreservation of in vitro neural samples at a high throughput fashion without CPAs or ice. The bedrock of this is what we're calling the NeuroVault pad or the NeuroVault well plate seal. And this essentially is this air-free system whereby we remove the air-water interface, sealing the individual wells into isolated enclosed systems using a compressible 3D printed silicone pad. And then we apply a solid backing, and that enables us to stabilize the supercooling state within that individual chamber. And because the volume of the individual wells is already so small, the thermodynamics of the system is super stable and much more resistant to the traditional sources of nucleation that we see from external forces such as vibration or ultrasound. We followed the traditional supercooling pathway for this. The only difference is that we found that when using neural cell culture, using a traditional hypothermic solution like UW is not ideal due to the high calcium or potassium concentration. And so what we're finding is that we can supercool just using the media, the cell culture media that the monolayers or the cultures are already living in, given that it's already at osmotic equilibrium and provides the nutrients and supplements necessary to keep the cultures alive, especially when they're reaching those low metabolic, the reaching states of low metabolism, and where they would traditionally end up in anaerobic respiration and start producing reactive oxidative species and see damage with calcium concentration influx. Um, the we plotted percent frozen versus temperature. So freezing within the well plate as a function of time and temperature, or freezing in the well plate as a function of the temperatures that we've surveyed. We found that -7 degrees Celsius was the cutoff for the well plates. So independent of the size, 6 well plates being generally the largest. When you get to -7 degrees Celsius, that's when you start to see partial nucleation within the plate in spontaneous individual wells throughout. But anything above that, -6 being the ideal temperature that we found for supercooling with this system, is completely stable for up to 2 weeks at a time. 2 weeks being the longest time period that we've currently tested with a living system inside of it. And that was using freshly harvested cortical brain tissue, thanks to our collaborators at the Miller Lab in the UC Berkeley Graduate Department of Neuroscience. As we can see in this figure over here on the right, this is the general schematic for, or the workflow for implementation of the system. You start out, and I can show you the compatible test beds currently. You start out with a neural tissue sample, that being either freshly harvested brain tissue or dissociated cells or plated adherent neurons, being stem cells, primary cells, organoids, spheroids, anything that you'd like, or a microelectro, like a brain on a chip microelectrophysiology system. So if you look for high throughput or functionality, that basically decides which pathway you're going to take in this process. And this is for adherent cell cultures. For tissue, as you can see with this figure below, you can just put the freshly harvested tissue inside of the wells into a 6 well plate, seal them independently, and then begin supercooling. But you take the culture vessel, you take our custom um, air-free insert, and when you put the two of them together and apply this enclosed cell culture system to a controlled rate cooling system, because the controlled rate cooling is a vital aspect to regulating the thermal stress and the osmotic and the metabolic damage to the samples. That's been a great base of work that Jasmine Velasquez from UC Berkeley has dove into with cardiomyocytes and is very, very easily translatable into other electrophysiologically active cells like neurons. Um, you end up with um, a supercooled cell culture system in a high throughput fashion based upon the idea that currently we don't have the means of inducing a stable supercooled state within a cell culture outside of deep supercooling or using a mineral oil to supercool, or a controlled rate freezer, which is usually very quite large and bulky and doesn't, you know, really handle the job in the minute ways that we need. Um, and so we've developed a platform for allowing um, cell culture vessels and the samples within those vessels to be supercooled stably for extended periods of time while also being resistant to the external forces inducing isonucleation, and all while not needing to introduce CPAs in order to stabilize the supercooled state. It's been wonderful working on this. Um, we've expanded into, at the moment, we have very solidly developed into the freshly harvested brain tissue as well as the multi-well adherent cell culture. So back up here, this first image, this is a 6-well plate with the sealing pad on it enclosed, and then you essentially just place a solid backing onto that, and that completely seals the plate and holds it tight. But the variations of this are really simple, and it's easily translatable and scalable to any combination of well or any well culture dimension or well number. So for instance, the image that's pictured there with the multi-well adherent culture is a 96 well plate. We can do 48, we could do 24, we could do 12, we could do 6. And then on top of that, because each of the individual seals themselves function, seals are functioning independent of each other for each well. If I can go back up a little bit, you see this is essentially just this, but singular. We can then translate this process into microphysiological systems. And my choice with this was brain on a chip systems, seeing as how currently the base of work on electrophysiology throughout or so during and as a function, a byproduct of the effects of on electrophysiology as a byproduct of the cryopreservation process has not been thoroughly explored, and definitely not from a circuits and systems neuroscience fashion outside of just tissue stabilization, architecture preservation, independent from the functional stresses that are, uh, being contributed to by ice nucleation or its CPA introduction. So essentially, this provides a system where it's very malleable to the user. You can introduce CPAs if you want to test out hypothermic conditions on large quantities of samples in an in vitro fashion, or you can straight away just go to the stable supercooled state and use it as a means of stable transport for in vitro samples. If we go a little bit further down, these are the problems that our system resolves. The main problems with cryopreservation of neurons is these four: the CPA toxicity, the dehydration by the solution, which is the osmotic imbalance, the formation of intracellular ice, and then recrystallization during warming. So traditionally speaking, if you're dealing with neural tissue samples and you want to preserve them, there's no way of holding them outside of the body because they degrade even faster than cardiac tissue and other muscle tissues, given the high concentration of mitochondria throughout the samples. Traditionally speaking, what's done in the industry is you take a freshly harvested sample, if we're dealing with just primary tissue, you fully dissociate via mechanical, enzymatic, or chemical dissociation, and then you introduce the CPAs and freeze it. And you do this so that the cell suspension is more able to be penetrated by the CPA concoction, traditionally DMSO, than it would be as a tissue, since brain tissue is essentially just a lump of fat. Very difficult to perfuse through, and to diffuse CPAs into, given that it's just all cell membrane, which is something that's quite overlooked fairly often, I believe, in my opinion. Um, but on the positive side, when we've tested the supercooled conditions on brain tissue, both as brain tissue samples freshly harvested and adherent samples, we got two interesting results. First and foremost, when we tested with adherent cell cultures in the multi-well plate, we found that I took the cell cultures and I used a micropipette to make cross hatchings on the bottom of the multi-well plate prior to the supercooling, essentially disturbing the functional network that had been established by the cortical neurons during their culture up to that point. So you can kind of see that in this where there's this track and the scratch on the bottom of the plate. But essentially, that creates a gap, and we can evaluate the functional connectivity and the capacity for the cell culture to bounce back from these disruptions in the network via evaluating their plasticity. And in that, it's neurite extension. So during supercooling, we see metabolic inhibition, which translates directly to developmental and functional inhibition. So when we pulled the sample out of supercooling, immediately we did imaging on it, and this tract was wide open. It was exactly the same as I had left it, undisturbed, no migration of soma, of the cell body into the region, no extension of neurites into the region to reconnect across it. But then at 24 hours, we see that the migration of the neurons into the open area begins, and the neurite reconnection begins as well. Basically showing that we get a developmental stagnation within our cell cultures while supercooling. Meaning that not only is this a wonderful tool for preserving fresh tissue samples or samples that can't be held in the incubator during transport, but also it's a wonderful method of safely inhibiting metabolism or regulating the developmental timeline of cell cultures, which is something that's very fundamentally important to neural tissue research in building in vitro models for neural systems, given that if you're using especially human-derived brain tissue or human-induced pluripotent stem cells, they age at the same time, along the same timeline as the human body, which means that before you can play around with the cells, you need to grow them and train them and develop them. And by a year of culture, they're exhibiting functional relationships with each other comparable to what I guess it wouldn't necessarily be a 1-year-old, but essentially the equivalent of a 1-year-old brain. Um, but the other side of that is that when we supercooled freshly harvested brain tissue samples, we found that we get a passive shedding of the glial layers, the white matter tracts within the brain that form the connective network between the cortical regions responsible for information processing. So when you look at the processing and use of primary neural tissue, um, traditionally speaking, you want the electrophysiologically active cells unless you're looking at glia or oligodendrocytes or something, the helper cells as a function of immunodeficiency or neuroinflammation. Traditionally speaking, if you're trying to understand the circuits and systems aspect of the brain, you want cortical tissue, hippocampal tissue, electrocharged tissue. The difficulty in this is that all of it is connected beautifully. That's what makes us special. But it's really difficult to isolate the glial layers from the cortical layers. And the cortical layers are postmitotic, meaning that they don't divide once they're in the culture and established in the brain. They have what they have, and there's no replication outside of neurogenesis in specific regions. Glial matter does replicate and replicates a lot in vitro. So when you're looking at cortical cultures, you want to isolate as much of the cortical cells from the tissue as possible, removing the glia. And usually that's done in the way that I screen for this, a very low level way of doing it is the capacity for binding is different. So glia are more likely to not bind immediately to the cell culture plate, meaning that I can't, or no, sorry, I apologize. Glia are more likely to immediately bind to the cell culture plate, whereas neurons need time to establish polarity and extensions in order to fully adhere, meaning that they need to kind of rest and settle into their space before they get comfortable and start growing. So a quick trick you can do when you're separating tissue is do an enzymatic or a mechanical or a chemical dissociation of the sample, and then you filter by weight. You essentially let all of the samples settle, and then after you incubate it for a little bit, the material that is bound to your well plate is predominantly more glial material. Um, and or pretty much entirely glial material, and the cell matter that you have left suspended in the well is predominantly cortical tissue with some glial tissue left, and then you can use, inhibitors for glial growth and astrocytic growth within your cell culture media, like B27, for instance. Um, but this all comes together, and I'm still taking a pipette and, chunking up brain tissue to then try and separate these things. But we found that with supercooling, we get a significant reduction in size of tissue. So the tissue itself shrinks, which I believe to be very heavily dependent on the osmotic equilibrium within the system. I think that using UW solution for brain tissue is a terrible idea, and we should transition away from it. So I think artificial cerebrospinal fluid or a media concoction with maybe some supplemental serums is probably going to be the way to go in the future. But we see that the comparison, if orange is the astrocyte count and blue is the neuron count, we're seeing fundamentally the same amount of neurons being harvested in the tissue, but significantly less astrocytes, significantly less glial cells within the cultures harvested. And because of this, we can isolate the cortical tract without actually dissociating or damaging the tissue in the process, maintaining the structural architecture of these very important brain regions, and also opening the door for glial isolation with cortical tissue stable, remaining stable superstructurally, and then that being processed as individual samples outside of the tissue as a whole. So we're seeing a defined cell type, different responses to the supercooled state and the supercooling process, the cryopreservation process. If we don't introduce ice, if we don't introduce CPAs, if we maintain an osmotic equilibrium, there are tangible differences between cell types, even within homogeneous tissue constructs. And so finally, just to kind of sum everything up, this is what I put together as a comparative overview of preservation methods for in vitro samples. The mainstay of this is cryopreservation. More recently, short-term cell pausing is a viable option with the development of novel perfusion apparatuses or better solutions for maintaining, cell or tissue structure at a above 0 degrees but low metabolic rate. Um, and when you put the two of these together, you get a pretty coherent view of what the field has. You have a form of long-form banking that requires ice, requires CPAs, leads to cell death, but the time span for which you can store things, the shelf life is so immense that it kind of negates the downsides in a lot of ways. And ideally, this would be, vitrification is the gold standard, but for now, standard cryopreservation. The alternative being cell pausing, which is a more metabolically, a much more metabolically active method of preserving samples for a much shorter time period, but you don't get the ice damage that's inherent within the samples. And the thing that's I feel is very important to note, there's a lot of discussion regarding, is ice actually a negative part, or if we localize the ice formation using CPAs, will that help mitigate the damage to the tissue? And if we can mitigate some of the damage to the tissue, is it worth, introducing ice in order to just do freezing, having some of that structural damage, and then kind of working off of that and still using the tissue? It's my opinion, having a background in neuroscience, molecular cellular neurobiology, and doing cryopreservation work from that perspective, that the structural identity, the architecture of the brain tissue is it is what makes it special. If you're disturbing the structural identity of the brain tissue, then you're not actually preserving the sample in the way that you're supposed to be preserving it. You're introducing crystallization that shreds the axonal connections, completely disrupts the tissue organization, the layering identity, the cortical column. There's so much that goes into the disruption of this system that if we can build away from trying to manage ice formation within the system and towards mitigation, there is a wealth of information out there that has shown us that functionally, if ice is not forming within the sample, then structurally there's nothing changing. If we can regulate the osmolarity, then ionically there's no, there's a minimal redox damage. Um, and if those two things are controlled, then we have a structurally stable, electrophysiologically, meaning functionally active tissue sample that we can preserve for extended periods of time. And that was the basis that went into the development of this system, air-free supercooling. I kind of highlighted which steps were taken from each side. So harvesting the sample is more akin to the cell pausing method. You don't necessarily need to dissociate in the way cryopreservation forces you to. You have to dissociate cell and tissue samples. You don't necessarily need to for the cell pausing, you can, but the main difference between the two of these is that for the air-free supercooling, we can do adherent cell cultures, suspended cell cultures, organ on a chip, pretty much any cell culture or any in vitro test bed that you can imagine, we can apply our supercooling method to in order to induce a stable supercooled state for weeks at a time. Sealing the wall plate is something that's unique. It's the most akin to the isochoric technique developed at Berkeley by Professor Boris Rubinsky and Dr. Matthew Powell-Palm. But ours differs in the fact that this is not an isochoric system. This is not a closed volume or a constant volume system. This is a means of removing the air-water interface in the same way that applying mineral oil might do, but in a much more stable fashion because instead of then remaining in an isobaric state, you enclose the sample to create a pseudo fixed, an enclosed environment, while still allowing for expansion. So the volume is not fixed. The pressure can increase, or the pressure technically can increase, but there would be no means of pressure increase via ice nucleation because it's such a stable state. But then you can see we introduce supercooling, which is more on the cryo side because it's sub-zero. So the degree of metabolic inhibition goes even further. I believe I saw a figure yesterday during Dr. Kim Young Dok Kim's presentation that that showed that the rate of metabolism is going down by about a factor. I think it was a factor of 10 every, or by half every 10 degrees. So if we're looking at a metabolic rate of, I would assume 7-ish at around 5 degrees Celsius, which is what is normal for the cell pausing temperatures, then we'd be seeing even at around 3.5, I believe, when you start getting into the -4 to -6 territory, which is a significantly higher degree of metabolic inhibition, and which is what we want because the less metabolism that we have for the cell's functioning, the more stagnation we get developmentally, and also the less damage occurs because the cells are not in a fasted state while also not having access to nutrients and not having a metabolic activity level required to produce more ATP or keep themselves ionically balanced. So pros for this, ideal for short-term storage or transport. It's perfect for taking a cell culture system and shipping it across the country. Ideally paired with some phase change material, something that can regulate the temperature without needing an external power system. If you controlled rate cool it to -6 degrees Celsius and put it inside a phase change box, ship it, it'll be stable for days, if not weeks. And then the only con is kind of just a byproduct of the cell pausing, is the metabolism still functions, but at a very low level, as we just discussed. But it combines the best of these two worlds into a more unified method of short-term cell pausing while also being hypothermic. Expanding the cryo supply chain. The idea behind this is that right now there's a lack of standardization within the field of neuroscience when it comes to the development of these cultures. Most people either harvest at their own site or just buy a cell suspension. And the cell suspension is traditionally promised at around 50% viability, 50 to 70%, depending on how your thawing process goes. So you're paying for only half of what you're actually getting. Um, oh, no, my bad. You're paying for double than what you're actually getting. You're only getting half of what you paid for. But on top of that, everybody's self-optimizing. There's a lack of unity within the field in developing these model systems. And so the idea is that if you introduce a means of of um, expanding the access to brain tissue samples in vitro on these brain on a chip systems or to freshly harvested tissue, you now have a reliable way of controlling or of expanding access to neuroscience research at the in vitro level, but also you open the door for more industrialized versions of cell tissue organoid manufacturing, where you can fabricate these in vitro systems in a standardized fashion at a center or a more industrial organization site, and then ship it out to your consumers. So this is we're very excited to announce this. This has been a fun year to work on this, and yeah, thank you so much. Huge acknowledgments to Kate Henn and the Miller Lab at UC Berkeley Neuroscience for the tissue, for the burst imaging, for all the help in the process. Thank you to Dr. Xiao Shan and Professor Gazzola at UIUC and the NSF Expedition in Computing. Huge thank you to everyone at ATP Bio. Wonderful community. Really appreciate all of the mentorship and the um, time spent. And yeah, thank you to everyone in the Rubinsky Lab. Made this possible. That's Anthony, that's Alan, that's Vivian, and Professor Rubinsky. So thank you so much for listening. I hope you enjoyed. Please feel free to reach out to me if you have any questions. You can comment it or you can feel free to email me. Yeah, hope you enjoy Cryo. Have a great day.